Photosynthesis is a vital process that converts light energy into chemical energy used by plants to produce food. One of the key light-dependent reactions in this process is photophosphorylation, which can occur in two ways: noncyclic and cyclic photophosphorylation. Let's explore how these two processes take place inside the thylakoid membrane. Noncyclic photophosphorylation involves both photosystem 1 and photosystem 2. Let's go through the process step by step. Light energy, or photon, is absorbed by the photosynthetic pigment of photosystem 2. The light energy is then transferred from one pigment molecule to another until it reaches chlorophyll A P680 at the reaction center of PS2. At this point, two electrons of P680 are photoexcited to a higher energy state. These photoexcited electrons are transferred to the primary electron acceptor. This creates an electron deficiency in P680 of PS2. The electron loss is replaced by electrons from the photolysis of water. During photolysis, water is split into two hydrogen ions, one oxygen atom, and two electrons. The oxygen atom then combines with another oxygen atom to form an oxygen molecule. The hydrogen ions are released into the thylakoid space. Next, both photoexcited electrons are passed from the primary electron acceptor of PS2 to PS1 via the electron transport chain, ETC. The ETC consists of plastoquinone, cytochrome complex, and plastocyanin. As the electrons pass through this chain, energy is released. This released energy is used to synthesize ATP from ADP and phosphate by chemiosmosis. At the same time, light energy is also absorbed by the photosynthetic pigment of PS1. This energy is transferred from one pigment to another until it reaches chlorophyll A P700 at the reaction center of PS1. Here, two electrons of P700 are photoexcited to a higher energy state. These photoexcited electrons are transferred to the primary electron acceptor. This creating another electron deficiency in P700 of PS1. The electron loss in P700 of PS1 is replaced by electrons that come from P680 of PS2 through the first electron transport chain. Then, the photoexcited electrons from the primary electron acceptor are passed through a second electron transport chain consisting of ferredoxin. Finally, the enzyme NAD+ reductase catalyzes the transfer of two electrons from ferredoxin to NAD+. NAD+ reduced to NADPH. Now, let's look at cyclic photophosphorylation, which involves only photosystem 1. First, light energy, or photon, is absorbed by the pigment molecules of PS1. The light energy is transferred from one pigment to another until it reaches chlorophyll A P700 at the reaction center of PS1. The electron of P700 is then photoexcited to a higher energy state. This photoexcited electron is transferred to the primary electron acceptor. This creates an electron deficiency in P700 of PS1. Next, the photoexcited electron is passed from the primary electron acceptor to the electron transport chain, ETC. The ETC consists of ferredoxin, cytochrome complex, and plastocyanin. During the transfer of electrons through the ETC, energy is released. The energy released is used to synthesize ATP from ADP and phosphate by chemiosmosis. Finally, the electrons from plastocyanin return back to P700, completing the cycle. In summary, noncyclic photophosphorylation produces ATP, NADPH, and oxygen, while cyclic photophosphorylation produces only ATP. Together, these processes ensure a continuous supply of energy molecules needed for the Calvin cycle and overall photosynthesis. That's all for now. Thanks for watching and keep learning.